the history of humanity is intertwined with the relentless battle against infectious diseases. Among these formidable foes, viruses stand out as particularly enigmatic and deadly adversaries. One such viral threat, lurking in the shadows, is the Marburg virus. This highly lethal pathogen, capable of causing severe hemorrhagic fever with alarming fatality rates, demands our attention and underscores the importance of vigilance in the face of emerging infectious diseases. Marburg virus disease, a severe and often fatal illness, is characterized by its rapid onset and devastating impact on multiple organ systems. The virus, a member of the Filoviridae family, shares a close genetic relationship with the Ebola virus, another notorious hemorrhagic fever agent. Understanding the intricacies of this virus, from its transmission pathways to its pathogenesis, is crucial for developing effective prevention and control strategies. The global health community has learned valuable lessons from past outbreaks, but the threat of Marburg virus remains. This exploration of Marburg virus aims to shed light on the virus's history, recent outbreaks, clinical manifestations, and the ongoing efforts to develop treatments and vaccines. Hemorrhagic viruses wreak havoc on the human body by disrupting the intricate system of blood clotting and circulation. Our circulatory system relies on a finely tuned interplay of clotting factors and blood vessels to transport oxygen and nutrients while preventing excessive bleeding. Hemorrhagic viruses disrupt this balance by infecting and damaging endothelial cells lining blood vessels. This damage can lead to uncontrolled bleeding both internally and externally. Internal bleeding can affect various organs, including the liver, kidneys, and brain, leading to organ failure and, in severe cases, death. The severity of hemorrhagic fever varies depending on the specific virus, viral load, and the individual's immune response. Some hemorrhagic fevers may manifest with mild symptoms, while others can progress rapidly, overwhelming the body's defenses. Prompt diagnosis and supportive care are crucial for improving survival chances in individuals infected with hemorrhagic viruses. Hemorrhagic fevers are caused by a diverse group of pathogens belonging to different viral families. This diversity underscores the complexity of these diseases and the challenges in developing universal prevention and treatment strategies. Each viral family has unique characteristics, modes of transmission, and geographic distributions. Among the viral families associated with hemorrhagic fevers, 4 stand out: Arenoviridae, Bunyaviridae, Filoviridae, and Flaviviridae. Arenaviruses, such as Lassa virus, are often transmitted to humans through contact with infected rodents or their excreta. Bunyaviruses, including Crimean Congo hemorrhagic fever virus, are primarily transmitted through ticks and can also spread through contact with infected livestock. Flaviviruses, such as dengue virus and yellow fever virus, are typically transmitted through mosquito bites and can cause a range of illnesses, from mild fever to severe hemorrhagic fever. Filoviruses, the family that includes Marburg and Ebola viruses, are among the most notorious and deadly hemorrhagic fever agents. These viruses are highly contagious and can spread through direct contact with infected bodily fluids. Marburg and Ebola cause severe, often fatal hemorrhagic fevers. Named after their discovery locations, these viruses have caused sporadic outbreaks over the last 60 years. Marburg was first identified in 1967 in Germany, and Ebola in 1976 near the Ebola River in Congo. Both viruses are filamentous, with single-stranded RNA genomes. Despite similarities, they have unique genetic sequences and antigen profiles. The year 1967 marked a turning point in virology. A mysterious hemorrhagic fever emerged in Marburg, Frankfurt. This event was the first Marburg virus outbreak, which shocked the scientific community. The outbreak was linked to lab workers handling tissues from African green monkeys. Workers developed severe illness with fever, headache, and hemorrhagic symptoms. Scientists raced to identify the novel virus, later named Marburg virus. The 1967 Marburg virus outbreak presented an opportunity for scientists to trace the virus's origins and understand its transmission pathways. Investigations led researchers to Uganda, where the laboratory primates had been originally captured in the wild. Further studies revealed that the natural reservoir of Marburg virus was the Egyptian rosette bat. These bats harbor Marburg virus without showing signs of illness. They often roost in large colonies, providing opportunities for viral transmission. Contact with infected bat droppings, urine, or saliva plays a role in transmission. Laboratory workers exposed to the virus through infected monkey tissues became the first human cases. This highlighted the interconnectedness of animal and human pathogens and the threat of zoonotic diseases. Since its dramatic emergence in 1967, Marburg virus has caused sporadic outbreaks, primarily in sub-Saharan Africa. One significant outbreak occurred in the Democratic Republic of Congo, with 154 cases and 128 deaths. Another major outbreak in Angola resulted in 374 cases and 329 deaths. This demonstrated the virus's potential for rapid spread and high mortality rates. More recently, cases have been reported in Uganda, Guinea, and Ghana. These outbreaks underscore the importance of strengthening surveillance systems and enhancing laboratory capacity. In 2021, a case in Guinea marked the virus's first detection in West Africa, which began in a rural village. The index case, linked to a bat cave, showed symptoms like fever and fatigue. Despite tracing efforts, the virus spread, resulting in 4 cases and 3 deaths. In 2022, cases in Ghana marked the first detection there, again linked to a bat-infested mine. In September of 2024, 6 deaths related to Marburg virus were reported in Rwanda, with 22 cases reported since the virus was confirmed. Most victims were healthcare workers. This has led to intensifying tracing, surveillance, and testing, with the hope of containing the spread of the virus. The recent Rwandan Marburg outbreak highlights the dangers of emerging infectious diseases, including hemorrhagic fevers, and the continued need to maintain vigilance. The primary animal reservoir of Marburg virus is the Egyptian rosette bat, a fruit-eating species in sub-Saharan Africa. These bats harbor the virus without illness, making them asymptomatic carriers. Human infection occurs through contact with infected bats or their excretions. Miners, guano collectors, or tourists in bat-inhabited caves are at increased risk. Contact with bat droppings can introduce the virus through mucous membranes or skin breaks. Marburg virus spreads efficiently from person to person, driving outbreaks. Direct contact with infected bodily fluids is the primary mode of transmission. This includes blood, vomit, diarrhea, urine, saliva, and semen. Healthcare settings are particularly vulnerable due to frequent contact with bodily fluids. Marburg virus triggers severe hemorrhagic fever. It rapidly replicates, spreads, and disrupts vital organs. Initially, the virus targets immune cells like dendritic cells and macrophages, which migrate and spread the virus through the body. After the initial infection with Marburg virus, a period of deceptive calm ensues, known as the incubation period. The virus silently multiplies within the host's body, establishing a foothold before announcing its presence with overt symptoms. The incubation period ranges from 2 to 21 days, with an average of 5 to 9 days. Infected individuals may remain asymptomatic or experience mild, non-specific symptoms. Early manifestations include fatigue, malaise, and a slight elevation in body temperature. These non-specific symptoms pose a challenge for early diagnosis, often leading to delays in seeking medical attention. As the incubation period ends, Marburg virus progresses to a severe phase with rapid onset of debilitating symptoms and alarming hemorrhagic manifestations. This stage begins around day 5 after infection. Fever escalates dramatically, often with chills, rigors, and severe headache. Muscle and joint pain intensify, leaving the individual weak. Symptoms may also include nausea, vomiting, and diarrhea, often with abdominal pain. Hemorrhagic manifestations emerge, a defining characteristic of Marburg virus. It infects and replicates within endothelial cells, which line our blood vessels. In so doing, the virus severely damages blood vessels, causing internal and external bleeding. In severe cases of Marburg virus disease, the illness leads to life-threatening complications as the virus wreaks havoc on multiple organ systems. The body's defenses struggle to contain the damage, often leading to a downward spiral. Hemorrhagic manifestations worsen, leading to significant blood loss and a drop in blood pressure. This can result in hypovolemic shock, a life-threatening condition. Multiple organ dysfunction syndrome is a common complication. The central nervous system is also vulnerable, leading to neurological complications. In some cases, Marburg virus can cause encephalitis, resulting in permanent neurological damage. The prognosis varies depending on several factors, including viral load and access to medical care. The case fatality rate is alarmingly high, ranging from 24% to 88%. In the absence of specific antiviral treatments for Marburg virus disease, medical care focuses on supportive care to alleviate symptoms and manage complications. Maintaining fluid and electrolyte balance is crucial. Severe vomiting and diarrhea can lead to rapid dehydration. Intravenous fluids help restore hydration and blood pressure. While supportive care remains the mainstay of treatment for Marburg virus disease, the quest for specific antiviral therapies and vaccines continues with urgency. Several experimental therapies have shown promise in laboratory studies and animal models, offering hope for effective treatments for human cases. Despite best efforts, Marburg virus remains a constant threat. Rapid public health interventions are crucial for containing outbreaks. Early detection and isolation are key to outbreak control. Healthcare workers identify suspected cases based on symptoms and exposure. Suspected cases should be isolated in designated facilities. Contact tracing is essential for breaking transmission chains. Infection prevention measures also protect healthcare workers. Adherence to infection control protocols minimizes transmission risk, while safe burial practices prevent virus spread from deceased individuals. The Marburg virus reminds us of the vulnerability of human health to emerging infectious diseases. Its severe hemorrhagic fever and high fatality rates underscore the need for vigilance, preparedness, and solutions. The lack of approved vaccines and treatments highlights the urgent need for continued research. Investing in research for vaccines, therapeutics, and diagnostics is crucial. The global community must therefore remain vigilant in detecting and responding to Marburg virus outbreaks. Thanks for watching. If you enjoyed this video, please subscribe to Science Curious.